Nicholas Eriksson from 23andMe, telling us about the future of genomic research, which of course is going to be crowdsourcing.
Okay, thanks. So I have, I guess, a number of confessions to make to start. So I don't know what crowdsourcing is, but it sounds cool. So I guess we do it. So I guess the other confession is about population genetics. I've always kind of felt, population genetics, it's a field of, pretty smart people working on relatively interesting problems with just absolutely no data, right? And, this talk is going to be exactly the opposite of that in all, well, hopefully not in all three directions, but it's going to be a bunch of simple questions that you can answer with a very, very large amount of data here. The other confession is that this is really, because I wanted to talk about new things that kind of no one up in Berkeley had heard about before. This is essentially all other people's work, and and much of it is kind of half-baked at this point. Okay, so just start out. I feel kind of an obligation coming from 23andMe to talk for just a second about risk and genetics, disease risk, and so on. So right now, if all of you in the room got genotyped, something like half of you would find out one of these, perhaps depressing, perhaps empowering, things on this list. If more than half if you're Ashkenazi, less than half if you're Asian, probably. But this sort of order of magnitude. 20% of you will find out that you are at 3 times increased risk of developing Alzheimer's disease, for example, and that those effects are probably showing up even in your 30s, 40s already. So this is right now. This is actually probably even about 2 years ago. This was true, and 5 years from now, this will not be 50%. This will be 80, 90, 99%, right? Okay, so that is, I guess, a brief glimpse of the future, and I'm going to make 4 very, very, very controversial assertions, I guess. So this first one. So what happens in GWAS? You find a lot of signals of very small effect. You find a lot of signals that all have odds ratios of about 1.1. And then someone says, well, 1.1, that doesn't mean anything. That's increasing my risk by 10%, whatever. That's the same as me going out and having a slice of cake during break. And actually, there's a Stanford professor who said exactly that. I won't mention his name, but. Anyhow, so it turns out that if you add a bunch of small things together, you multiply a bunch of these odds ratios together, you can get something that is big. Okay, so that's if you have 100 SNPs that have all been found associated with the disease, they all have an odds ratio of 1.1. You have 20 more risk alleles than non-risk alleles. You could be at pretty high risk. Okay, so the second point is if I gave you risk predictions across 40 different diseases, say. Essentially, everyone will be in the top 5% of one of those, predictions, right? We're all at high risk for something. The third point, I guess is kind of directed towards the FDA, but people deal with a lot of kind of crappy things in life. Not always with the medical doctor's help. And then this is actually live on the internet, isn't it? Sorry. Sorry. So the last point here is that okay. So you can't be changing your DNA or so on, but you do have the nice interpretability here of the DNA is kind of clearly causal. These SNPs. Up to some details, they're actually causing the disease. The disease is not causing these different alleles. So this is both better and worse than traditional epidemiology, where high cholesterol and heart attack risk. Well, this is is it truly causal? Is it not? I mean, it's certainly something you can intervene on, but is intervening on it going to help, and so on? Okay. So so 4 points about risk. And we talk then just a little kind of keep things very, very light at first. So it turns out for the Olympics, we asked a bunch of people what sports they played, and this is the lines show up a little bit. This is a spectral clustering of the correlation matrix between what sports people play. So These are not my labels, but you've got the you've got the the weightlifting and the elliptical machine cluster over here. You've got the young people sports. So it turns out you actually pull out a lot of structure just by asking a lot of people a question and doing something very dumb there. So then it turns out we've got something like 25,000 trios in the 23andMe database. And so we looked, again, for Valentine's Day, we looked at what factors are making people couples. So we looked at HLA similarity and found kind of completely null results there. If you look across different phenotypes, it turns out that couples tend to both be punctual, they both be skiers, hikers, both be non-smokers, fast food lovers, or both be kind of apology prone. But there were a couple of traits that go in the opposite direction. So it turns out that people who wake up early and people who go to bed late are tend to be attracted to each other, or perhaps this is I don't know the direction of causality. Perhaps after you get married, you start to avoid your spouse. Yeah. So okay. Okay. So but then the interesting question is what happens with your with your kids, right? So so heritability is kind of the I mean, especially when you have kids and you're trying to assign proper blame for things between you and your spouse, you'd like to know what to do. So this is joint with Breland Brown, who's Leo Proctor's student here, Aaron Kleiman, Nick Ferlat, Emma Pearson, who are all at 23andMe. I should mention that, so Emma Pearson, we are unfortunately going to lose to grad school. She is terrifyingly smart, and I would like her to find a good home, actually. So, yes. So if there is someone in computer science who would like to try and convince her to come, please let me know. Okay. So we've computed heritability numbers. These are heritabilities across various disease traits. So this is cut off a little bit, but you have pigmentation traits, you have immune traits like allergies, you have food preferences, various metabolic traits, cancer traits, morphology, hair, neurological, on and on and on. So these are heritabilities in Europeans, computed with GCTA, actually. So this is heritability tagged by common SNPs. And you do see, so somewhat the pattern that you would expect. Your pigmentation traits are very heritable, so on. It turns out that if
Quick question.
Yeah.
Are these traits self-reported on a form?
Yes.
Thank you.
Yes. Okay. So if it turns out, so I've been working for 5-6 years now, trying to figure out how to give an interesting talk about GWAS, other than here's some SNPs that are associated with the disease. So I'm going to kind of entirely skip that. It turns out that more heritable conditions we find more hits for. So these are the number of significant associations in the 23andMe database, I guess square root to make it look a little bit more linear, and the more heritable traits such as height, eye color, for example, see a lot of hits. You can see, although this is somewhat confounded by sample size, you can see some difference in kind of genetic architecture, right, where height is kind of known to be the sum of a lot of very, very small effects. Eye color, there are a handful of SNPs here that explain a very large percentage of the variance in eye color. So we've been doing this across populations. So most of the results aren't ready, unfortunately. These are the however, in blue these are the African heritability estimates. In red these are the European heritability estimates. You see a few differences, but they are mostly so. This is not quite readable, but this is freckling. This is hair color. These are two of the biggest differences. And this is probably not a real difference, but simply kind of a problem with interpreting heritability in an admixed population, right? So among African Americans, your freckling propensity, your hair color is really determined entirely by how much European DNA you have, right? Okay, so. Okay, these are a few things that we've been working on this morning. So the broad idea here is that we're trying to compute heritability across hundreds of different traits in a few different ways, all with different biases. And in particular, one of the things you can do is take all of the trios. You can get a heritability number there, and that heritability number is going to include your shared environment, right? So, if you just look at the correlations between the kids' traits and the parents' traits, that is a heritability plus shared environment estimate. And there are a bunch of things that we're kind of trying to puzzle out here, which I assume are mostly due to environment, mostly due to the shared environment, but I kind of can't explain all of them. So for example, for daughters, their BMI and their height are much more strongly correlated with their mother's height and BMI than their father's. And for both sons and daughters, their diet and political views are more strongly correlated with their mother's diet and political views than their father's. And the opposite for smoking. So most of these you can kind of squint your eyes a little bit and say, okay, this makes sense with shared environment. I don't know how to explain the height result. It seems to be significant. It seems to be so I don't know if there's an actual possible genetic mechanism here, or if or what's going on with that. But ideas would be helpful. Okay, so now talk. Have a little bit of math in this talk. So this project, this is with Eric Durand and Corey McLean. It started off actually when we started trying to use long-range phasing to phase all of the 23andMe data set. So we're now at 600,000-700,000 people in the data set. About 90% of them are European, and so at that at that number, if you compute IBD down to say 2 centimorgan segments, you actually expect to have enough segments so that every person will have kind of surrogate parents at every point in their in their genome, enough so that you can essentially perfectly phase them. So it turns out that our long-range phasing algorithm worked really well in simulations and really poorly in practice. So we started to ask why that is, and so should just say this. So So if you have people that are computationally phased, you probably have say a switch error every centimorgan, right? And so you are looking for IBD segments that are much longer than a centimorgan. So you're likely to see switch errors in there. So you're likely to see some pattern like this. And if you're running IBD on 500,000 people, the only in any way feasible way to do this is to use GERMLINE, right? You cannot you can't be using Beagle. It's just kind of impossible. So what GERMLINE is doing, right? In its its mode here is it's taking all of these blue segments as seeds if they're long enough, and then it's sliding along and trying to extend them unless it hits an opposite homozygote. And if you get a long enough segment there, you report it as a potential IBD segment. Right, and so it can go through and correct those switch errors. So what we did is we looked in trios, and the trivial observation, right, that if you have an IBD segment between the kid and some other person, then it better be IBD between either the dad or the mom and that other person. Okay, so what we did is pulled we actually only used something like 10% of the trios because there was so many IBD segments. But okay, so we pulled 3,000 trios, some 17,000 Europeans, ran GERMLINE and everybody in this, and so I should say everyone here is just computationally phased with Beagle. So it turns out you get a lot of segments. Most of those segments are short. There's a total of something like 13 million segments. This was only on a couple of chromosomes. And our measure of correctness here was just how many informative sites are shared between the IBD segments, right? So here this brown segment is the child other IBD segment, and similarly this mother segment and this father segment are the mother father other IBD segments. So here the mother has kind of five shared ones out of 9, I guess, and so here we give this segment a score of 55% of 55% overlap there. Okay, so that said, if you look across all the about 13 million child other segments that we found on these chromosomes, well, a lot of them show very, very little overlap. So 61% of them show no overlap at all. So there's no parental segment that is that's overlapping at all. Another 25% are showing a little bit, and 14% of them seem really good. So if we look at these truncated truncated segments and go back and see are these actually good calls, we see that we're calling the opposite homozygote that is truncating this segment, we're calling it with high confidence almost all of the time. Oh, sorry. Yeah.
This may be silly, but you had already checked that these trios were good trios, like the father was who they were supposed to be.
Yeah, these were trios detected by IBD. Yes, okay. And there was yes, they were. I don't know if they knew they were trios, but they were trios. Yeah. Okay. But so it's not a case of genotyping error causing false negative parent IBD here. And so also if you look at these segments that show no overlap, well, they almost all show a lot of opposite homozygotes in the parents, right? So we're really confident that these that this measure is correct, and that we're not actually seeing a ton of false negative parent IBD. And this this kind of correlates with how GERMLINE is tuned, right, to give you to find a lot of segments of of which not all of them might be perfect. So we so this is average overlap, and is this cut off? Sorry. Okay, sorry. So you see that segments above 4 or 5 centimorgans, they are almost all red, which is essentially kind of perfect overlap. But the average overlap for all of these 2 to 4 centimorgan segments is very, very low. And there's some artifacts, because we're doing this on small chromosomes. So you see the segments going through the centromere. And
what's the cause of the stripe in the middle, in the reds?
This one, I don't know actually. So there's some filtering actually that went on before this. To throw out regions of low SNP density and regions of regions that just show these very, very big peaks of IBD. I'm kind of guessing that we missed a couple of those. Okay. So and we just chose 80% as kind of an arbitrary threshold for what is true. The results are robust. And so we wanted to score these a little bit more reasonably. So let me give you an example here, right? So if we have if we have these 2 individuals here, we could either say that that they share this segment IBD with a total of 3 switch errors across there. Or we could alternatively just say that there were 2 genotyping errors that were leading to this. So as you probably see where this is going, we're going to give a cost to to GERMLINE allowing a switch error, a cost to GERMLINE allowing a genotyping error. It turns out to really not matter much what the cost is, but here we went through an estimated switch error rate, a genotyping error rate. And gave this haploscore as sum of how many things that you are how many switch errors, how many genotyping errors you're requiring for this to be IBD. It turns out this can be solved efficiently with dynamic programming, which is probably something one should not go through late on a Friday afternoon, but this can be solved. This can be computed easily enough. So it turns out the nice thing is that this haploscore is very discriminative. So here the y-axis here is our computed haploscore on segment by genetic length. The red segments are the good ones. The blue ones are the bad ones. And you can see a really clear delineation here between good and bad segments, according to the haploscore. So theoretically, this line probably this curve here probably should actually be a line. There's a little bit of weirdness in the kind of 2 to 2.5 centimorgan range because of the fact that we detected everything with a 2 centimorgan cutoff, and then we're using this kind of 80% coverage feature, over and over. So really, I think our results should be kind of valid from 2.5 centimorgans up or something. But it turns out that you can efficiently throw out the millions of segments that are wrong. This works, but kind of this works on other chromosomes. This works in a few other data sets that we get. So okay, so we can do that with IBD. Let me talk a little bit then about about the history of America, I guess. This is Kasia Brick, Eric Durand, Joanna Mountain, mostly doing this. So the data set here is about 170,000 people who are living in the U.S. and who gave us self-identified ethnicity, so as either European, Latino, or African American. This set. So okay. So when I use any of those terms now, I'm going to mean self-identified ethnicity, and then we have a local ancestry assignment method that using either 25 or 26 different countries or regions. So these are the regions along with the kind of training sizes. These are trained using HGDP data plus a lot of 23andMe data from customers who reported having four grandparents who were Ashkenazi or from one of these given countries. So this local ancestry method works fairly well. I'm not going to talk about it, but it's interesting to see how that interacts with self-reported ethnicity. So these plots here, which hopefully are readable. These are for African Americans. So this is plotting up here. This is mean African ancestry by state of by state of birth. I think we have both state of birth and current residence, but I think all of this is state of birth. So you see that African Americans in the South, have higher average mean African ancestry than African Americans in California, for example. So here are the two distributions, and that there's a non-trivial proportion of Native American ancestry in the African Americans. Okay, so similarly, if we do this across Latinos, so this is the so this is the same plot of how much American ancestry, how much Native American ancestry you see for the self-identified Latinos by state, and also the total the projection down onto just European, African, and and Native American components, I guess, of the their genome-wide percent for the Latinos. And now the hidden part of this is looking actually at people who self-identify as Europeans, right? So here are these are the frequency of Europeans with at least 2% African ancestry, the frequency of Europeans with at least 2% Native American ancestry. And you see this is really quite high. So this tops out at about 5% in South Carolina and Louisiana. and similarly in the Southwest and well, I guess most of the West, for the Native American ancestry. So this means that so I think the total nationwide is something like 1.8% of self-identified Europeans in the 23andMe database with whatever biases that has. About 1.8% of those self-identified Europeans have a non-trivial bit of African ancestry. So 2% is Our local ancestry method is certainly not perfect, but 2% of the genome is a lot of the genome. Where that's not a false signal at all. You'll see also that this is not actually. This certainly correlates with the states. So the percent of Europeans with some hidden African ancestry, it correlates with the states with high African American populations, but it's far from exactly the same, right? So you see that Alabama, Mississippi, Georgia actually have much lower lower hidden African ancestry in the Europeans there than Louisiana and South Carolina. So okay, so mating is not done at random.
So there's lots of individuals from each of these states. So I mean, all the color differences I can pick up are pretty strong.
So the white states are well, I guess are the white states. Yeah. So I should have mentioned on all of these that we did actually throw out states where there wasn't enough data.
But Latinos in Mississippi. That was.
There are not many Latinos in Mississippi who are 23andMe customers. Yeah. Yes. So yes.
So
these,
I'd like to make a comment. That
Yeah,
your slide is of course highly political. It probably has political consequences. And I would just tell you a fact.
Okay.
That if you do genetic studies in South Africa, which is about as racially charged historically as you can possibly imagine, you find substantial numbers of South African whites with quite substantial African ancestry.
Yeah.
And I'm not sure exactly the connection, but I think there's something common.
Yeah. No, my wife's family is Uruguayan actually, and her parents claim that in Uruguay everyone is 100% European, and it is it is, which it's very obviously not. Yeah. So yeah. It's there's but this is a lot of people, right? This is 2% out of I don't know 200 million self-identified Europeans in the U.S. or something. So there's actually millions and millions of people out there with something a little bit surprising there. Okay, so we've been working, and I won't go into this into trying to date these admixture events. But this is all very, very fresh out, so I'm not going to really talk about this. Instead, talk a little bit about recombination. So this is a project with Adam Otten, his student Chris Campbell at Einstein, Nick Forlat at 23andMe. So we've, well, at the time these figures were generated, we had about 4,000 quartets across a bunch of different populations, and inferred recombination. Recombinations in all of them with Merlin. So we see, for example, here's the distribution of events per meiosis for maternal and paternal events. As you would expect, I think everything I'm going to show you here is well known, and this has essentially just been kind of our quality control check so far. But if you build a genetic map, we see the same map that that Decode did, up to a couple of changes that we're trying to figure out if we have errors or not.
Sorry.
Yeah?
Does it hold if you exclude the subtelomeric regions? Because it's been known to bias this kind of estimates.
I don't know if we've tried that. I didn't know that was known. Okay, we will have to take a look. Yeah, so as I said, this is very much there was a flurry of emails about this today as well. So these slides might not be totally up to date. So these are but if we look at hotspot usage, so again maternal and paternal hotspot usage, we see the higher usage in for the paternal events. This. And then I should say, if we run a GWAS on hotspot usage, we see PRDM9 quite clearly. So I actually really wanted to share a couple of other things that might be novel here, but we don't know if they're true. Sorry, this fell out. But anyhow, there may be some evidence of some differences with these various effects looking across populations. Which I believe those results should be novel. Okay, well, I'm going to end a little bit early perhaps, and one, take questions, but also say that we have more data than we can realistically deal with. We haven't been promoting this very heavily, but if you're good with Google, you can find this page here that where we're actually calling for academic collaborators. We've been reviewing these proposals a couple of times a year, in the spring, in the winter, and it's short, it's easy. If you have some questions that can be answered relatively, without a ton of work, but with a lot of data, we would love to have some new ideas to work with. I should then say thanks to a lot of this was supported by NIH, and of course, much more so by the customers who paid for the genotyping and answered questions and so on. We've got Corey McLean, Eric Duran, Joanna Munn, Nick Follett, Emma Pearson, Aaron Kleiman, all at 23andMe, Breland Brown, Kasha, Adam, and Chris also with the projects I mentioned before. So, thanks.
Can you just show that slide about hotspot usage in males and females again?
Yes.
So is that a difference were you claiming between males and females in hotspot usage?
Yes. We do see a difference. I mean, this is all conditional on the assumption that our data is properly cleaned up, which is not 100% true yet. But yes, we are seeing we are seeing increased hotspot usage in males. Yeah. And some differences across populations, but I'm less convinced about that because the sample sizes are not all large.
Do you have ancestry differences between mothers and fathers systematically?
I believe so, but I don't. It has been changing every time we've been writing it, so it's not quite clear. Yeah. Sorry. Yeah.
Along those lines, for the 25,000 trios, have you looked for any patterns in admixture or assortative mating, the ancestry based?
We have found we've found trivial things, I guess. But nothing non-trivial, I guess. Is, I don't know quite what the it's a difficult one. We come up in detail. Yeah.
I'm just trying to remember if I heard correctly. So when you did the couples thing for Valentine's Day, you said the HLA was completely null, meaning so was there an equal chance of having an identical type?
We saw no difference between HLA sharing between couples and randomly selected, permuted couples or different parts of the genome or anything else, really.
Are there location-based differences in the ancestry proportions actually, never mind.
Okay.
Is 23andMe still online accepting customers?
Yeah. No, you should sign up right now. It's $99. I mean, right now you're not going to get your health reports back, so you're not going to get the scary information that I told you about at the beginning, unless you are smart enough to look up. You can download your raw data and you can do with it what you may. But you get all of the ancestry stuff back. So.
I see. So once someone has access to the raw data,
Yes. This is the cheapest way of genotyping people, probably.
And you provide some sort of key, that like, do you provide any analysis tools for the raw data, or?
No,
Or
but
just
there are plenty of people who do. Yeah, and it's not so hard to do. Yeah. Yeah.
So having applied apple score, did you now phase the 600,000 individuals?
Yeah. So we're working on that. It's it cuts down the number of segments so much that we're that we're not seeing quite as much over. It looks like from our simulations that you need, I forget the number, but maybe something like 10 surrogate parents per position to really perfectly phase that.
On average or minimum?
For minimum, I guess. If you want to really assure that you're not going to get a switch error in that in that spot, then you need more than a few surrogate parents. You need something like 8 or 10. And I believe so. It hasn't been working really well, and I believe that's just because a haploscore is throwing out 2/3 of the data. So yeah.
Just to clarify, so your strategy is to run Beagle, then GERMLINE to get the IBD segments, and then you're going to do long-range phasing. And
then rephase everybody.
Okay.
And then re-impute IBD. And yeah, yes, that is what we're trying to do at least. Yeah.
I'm a bit biased, but I might suggest that ShapeIt might automatically do that for you in one step. I mean, the advantage being that the model actually, looks up how haplotype sharing of each individual to all other individuals in the given sampling.
Yeah, I mean, so we've had internal debates about exactly this. So one, I mean, it's slightly, licensing requirements have stopped us from using this kind of full scale. But there are two camps on this issue within 23andMe. We've done a little bit of testing, but it doesn't seem quite as clear as I had hoped from reading the ShapeIt papers. But we might be doing something a little weird. Yeah.
With the admixture of Africans into Europeans, I know you said that you didn't have any comment about the timing, but then I was sort of curious just in terms of, do you have an idea of is it something you're thinking in the last 200 years or so, or is this something like 10,000, 20,000 years ago?
This is actually something that Kasia sent me this morning. So you probably have as good of an idea of what this means as I do. Yeah, sorry. It's all a little bit last minute, but yeah.
So you said at the beginning that 1.1 to the power 20 is 6.5.
Wait, is that all right?
Well, I don't know.
Okay.
But the thing is, so in a lot of these GWAS studies, if you looked at the SNPs, some of them could have been on IBD blocks. And so then the question is whether the risk alleles are really separate risk alleles, or if they're actually just sort of implicitly inferring that you have that IBD block, and that IBD block has somewhere on it a causal SNP. So then you wouldn't multiply those together separately.
Yeah, it's definitely true that you have to get independence between these. But even even now you're ending up with a lot of GWASs having 50 to 100 entirely independent signals.
So in the health reports that are no longer available, I mean, how did you take care of that?
So almost everything we report on entirely independent SNPs. I think our melanoma report we do something a little bit more sophisticated, where there's two SNPs that are in pretty high LD that appear to have some independent effect that is not total, and we have a model that takes care of that. But I think there's only one or two examples of that.
I'm curious about what does the raw data look like, and how easy is it for an individual to figure out the meaning?
I wonder if I have it. I mean, the raw data really is, RSID, perhaps chromosome position in some coordinates, and then allele 1, allele 2.
Okay, so on the health report,
but yeah.
I did do a 23andMe $99 thing, and I got a report, but there were no error bars on the estimates of frequencies. Why was that?
Yeah, I've been slightly fighting for this for the last 5 years. It is true that there should be error bars. It turns out statistics is very hard to explain to people, and that statistics with error bar, I don't know. It's not user friendly, I guess is the short answer.
How do you do ethical, what's your ethical consultation for making these decisions on what to report? Who is in charge, or how does that work?
The FDA is listening.
God, you know, we have, you know, we do have a really great group of kind of medical geneticists who, you know, who are kind of guiding us in what is, you know, in making sure that we do things right. I think, you know, I'm a little bit biased here, but I think we actually err too much on the side of overcautiousness.
All right, let's cut it off there. Now, Yoon tells me there are no final announcements, so we can do this all at once. We can thank Nicholas again. We can thank a terrific... And we can thank all the speakers for a terrific set of talks at this meeting. We can thank the staff of the Simons Institute and of course, most important, we can thank you for really organizing this whole thing. He was like the mastermind behind it. It was his idea, and it was a terrific job. So thanks, you.